What I want to do today, sort of following up on what we've heard about sort of signals yesterday and sort of the signalers that produce them, is for the rest of the talk, focus our attention on receivers. And historically, when we think about receivers in a communication system, we often think about work related to how they interpret signals or get the meaning out of signals or the information out of signals to use that information to make adaptive behavioral decisions. And so what I'm going to talk about today is actually something that I would argue occurs sort of perceptually upstream from extracting the meaning out of signals, okay? And I want to stick with this example here for a second with the European starling. Of course, like other animals we've talked about, it has a signal that comprises a sequence that includes different subunits. I've labeled them U1, U2, and so forth here. And there's something else interesting about starlings beyond producing these sort of sequences, is that they often produce these sorts of sounds in very large, noisy social aggregations, not unlike humans, for example. And so in an environment like this, okay, the issue is not a sequence. The issue is an overabundance of sequences from the animal's perspective. So the animal here is suffering from sort of a biological analog of the human cocktail party problem, right, which refers to the difficulty we have perceiving speech in a noisy social environment. And so if we want to understand how receivers get meaning out of signals, we first have to understand how they construct or perceptually reconstruct the sequences that the signaler produced. Okay, so I'm going to talk about today how receivers go about perceptually reconstructing the right sequence in environments where there are lots of competing sounds. Okay? Now, the framework we've adopted in my lab for studying this is one initially sort of, formalized by Albert Bregman in his book Auditory Scene Analysis, published in 1990. And if you have an extra 2 months with nothing else to do, I suggest you pick it up and read all 1,100 pages or something of it. It's a very comprehensive, thorough book. And what auditory scene analysis in general refers to is just the perceptual organization of the sounds in the soundscape or in the acoustic environment. And Bregman devotes about a third of the book to discussing how humans organize perceptual sequences. And he refers to this as something called auditory stream segregation, which just refers to the perceptual organization of sound sequences into coherent or meaningful representations of sound sources or events in the environment. And he largely treated auditory stream segregation in the context of human language perception in noisy environments as well as music perception. And those are the two dominant fields in which auditory stream segregation has been considered. But let's think about our starling, right? This is an animal that's communicating in a noisy social environment, and there are lots of these units flying around. So what we want to understand is how the starling's auditory system perceptually binds the appropriate units together. Okay? And this is a process Bregman would refer to as integration. And at the same time, how that bird's auditory system segregates these correct units from incorrect units. And so Bregman talks a lot about integration and segregation as being sort of opposite sides of the same coin, if you will. Right? So when something gets integrated into a coherent auditory stream, it necessarily is segregated from other streams, right? This is something he referred to as the principal exclusive allocation. Now, in his book, Bregman goes into great detail about trying to understand how auditory systems might exploit cues in the sounds themselves that would sort of instruct the auditory system how to form the correct auditory streams. And he called these cues, or he pointed out how these cues relate to what he called environmental regularities. And what he really means is that they're related to sound production mechanisms and sound propagation. And he identified several different types of cues that auditory systems can potentially exploit in order to form auditory streams. And I won't go into the details here. There are several. I'm going to focus on these top two. But before I do, I'm going to interject a quote from Bregman. It's more recent than his 1990 book. This is something he wrote a few years ago. And I think it's an interesting quote. I'm just going to read it. He asks, do animals other than humans perform auditory scene analysis on auditory inputs, that is, the parts of the signal? And if so, do they exploit the same environmental regularities as humans do, or are there other regularities specific to the animal's own environment? Now, the reason I find this an interesting question is because, first, it's posed by a human psychologist. Second, it's written in the foreword to a book on computational auditory scene analysis. So it's written by an author in a context very far removed from what we think about. But I would further submit that the field of animal behavior, people who study animal communication, in general have not taken up this question yet. But I think it's an important question we should be asking, especially in the context of how animals analyze vocal sequences themselves. And so I'm going to stick with these two cues, and I want to just illustrate, if I can, with what will almost certainly be an ill-fated demonstration. But I want to play some sounds for you, some sequences of tones. And what I'm going to play for you is a target melody that's been interleaved with some captor tones. I want to try to illustrate how differences in spatial location and differences in frequency or pitch might be used to segregate an auditory stream that corresponds to the target signal from these captor tones. And so the melody is just outlined here. Okay? But now have a listen and tell me if you hear a melody. If you do, don't name it. Just tell me if you hear it. How many of you did a melody sort of pop out? Okay, write down what you think it is if you haven't heard this demonstration before. I'm just curious. Now, for many of you, you didn't hear any obvious melody, right? So in this next example, I'll play the same sound, except in this example, I've shifted the melody to come from one speaker and the captors to come from the other speaker. And I apologize for the limited spatial separation, but I'm going to call this a large one, just for convenience. So, do you hear the melody now? How many of you picked up some hint of a melody? Oh, Dan, you've lost the melody now. So that's hard, okay? Now, in this example, right, I'm going to shift the melody up in frequency range about an octave outside the range of the captors, okay? But they'll come from a single, well, there's no spatial segregation here. See if you can detect any sort of melody now. How many heard a melody that was maybe something like Mary Had a Little Lamb, we would call it? Yeah, something like that. So the point here, right, is that your auditory system is exploiting the frequency difference to hear the melody as a separate auditory stream than the captors. Another minor point I'll make here is that we all inherently think sound location is the answer to perceptual stream segregation, and the evidence suggests it's actually pretty weak cue. It could be completely overridden by other cues, and that was borne out in the demonstration here. I think a lot of people had some difficulty hearing this, the stream here, and that's typical for reverberant environments. Yeah. Sorry. No. So, with all of this in mind, okay, I want to move to the animal I'm actually going to talk about today. And so this is the animal we work on. It's Cope's gray tree frog. It's about the size of the end of your thumb. We can get in boats and follow them around sometimes, but we don't have to. We can just sort of walk right up to them. These males produce what I'm going to call a sequence of pulses. Okay? Each pulse is produced by a separate independent contraction of the trunk musculature. The pulse rate of the call is about 50 pulses per second. And I felt a little inadequate after seeing some of the talks the other day, so I went home and did an analysis. And so each of these pulses has acoustic energy at two different frequencies, about 1.3 kilohertz and 2.6 kilohertz, and I'll come back to that in a little bit. Now, this 50 pulses per second I'm going to emphasize is sort of important, right? That keeps coming up over the rest of the talk. But I want to sort of make a more general comment here about how the sequences of these elements, of these units, with pulses, right? They take on behavioral relevance that any single unit of production lacks by itself, okay? And we know this from decades of work by Carl Gerhardt and his students and collaborators over the years that have looked at the the meaning, if you will, of various acoustic features of this signal. For example, this pulse rate turns out to be a really important species recognition cue. Females strongly discriminate against slower and faster pulse rates. The number of pulses in the call turns out to be a feature that's favored by female mate choice, so it's subject to sexual selection. The total number of pulses males produce per minute over long periods of time, also something females can cue in on. And the point I just want to sort of make here is that none of these properties of the signal emerge without the animal integrating the pulses together, right? Integration has to happen for these sorts of things to be perceived. Now, the reason I'm sort of making a point about this is because the acoustic environment in which these frogs reproduce is quite a mess. And so here's a typical acoustic recording made at the position from where a female might make her decision about mate choice. And so the call itself sounds like this. And this is the acoustic environment in which receivers have to perceive that call. So no. They actually do quite well. What I've actually shown here, believe it or not, is actually a simple example, a relatively simple acoustic environment. This is only one species calling. Now, across most of their geographic range, certainly in Minnesota at certain times of the year, they're also breeding at the same times and in the same ponds with these other species, all of which produce pulsatile calls. So mate choice occurs in this environment sort of awash with pulses. Right? And so I want to understand how the frog's auditory system integrates pulses produced by one signaler, right, and segregates those from other signalers in the environment. And I'll just sort of very quickly sort of gloss over some of the methods we use to look at these sorts of questions. The behavioral response we're interested in is phonotaxis. And so you bring a female frog into the lab, put her in an arena. So this is a 2-meter diameter arena here. You play sounds from a speaker that imitate the call. They exhibit this robust approach response, which I can sort of illustrate here. And I think you should be able to see this. The female is being released from a central release cage here. There's a speaker hidden behind an acoustically transparent wall. So a fairly unambiguous behavioral response. And so all the experiments I'll talk about today involve phonotaxis, and they involve manipulations where we present either multiple sounds or we have multiple sound sources where we're interleaving pulses and asking the animal, do you hear this as a signal, or is this heard as something else? Okay? So I'll tell you quickly about 3 experiments we've done. The first of these was simply looking at spatial location as a grouping cue for auditory stream segregation. And so what we did was take 2 different sequences, each of which had the wrong pulse rate. Okay? By themselves, females don't like this sound at all because it actually corresponds to the pulse rate of another species that they can't hybridize with or anything very easily. And so what we did was present these 2 sequences in an interlead fashion from either the same location or from different locations. And we asked, well, if they integrate them together, they should hear something that's attractive. They should exhibit this robust response I just showed you. And if they don't, they should ignore it, right? Because it's the wrong species. So we did these experiments, and one of the key response variables we often measure is latency. These females exhibit very short latencies, around 50 seconds to travel a meter to a very attractive signal. And what we found is that when the 2 sounds came from the same source, they found it very attractive. And as we separated the 2 sources, the response latencies went up, which was perfectly consistent with our initial interpretation, which was, well, they don't like this sort of sound. But then we did this really foolish thing and sort of dug a little bit deeper in the data. And we started looking at how far these animals were moving in the arena. And what we found was this direct correlation between response latency and how much the animals were moving. Now, that could have been because the animals were just sort of wandering around. They weren't interested in the sound. But that's not what happened. when we looked really closely, we found responses that look like this. Okay, so these are just traces of the phonotaxis paths in these different conditions. This is one female across all tests. And at low spatial separations, they pretty much went to a speaker, right? But something interesting happened when we started separating the speakers even further, right? They would initially approach one and stop. Turn to approach the other. This is really clear in the 180 degrees, right? And these females would just go back and forth in the arena. So what that rules out, especially along sort of an axis between the speakers, that they weren't having any difficulty localizing the signal. Right? They're aimed at the signal no matter what they do. What they were actually doing, we think, is integrating pulses over a large spatial separation. So by the time they got here, right, this was no longer attractive. But suddenly the attractive sound sounded like it was coming from the other side of the arena. But the only way that could be the case is if they integrated the two pulse streams. Right? So our interpretation of these data for now is that spatial location is a pretty weak cue for stream segregation in this context. Now we moved on to do a few other experiments. Here's one looking at frequency similarity. This is sort of the Mary Had a Little Lamb experiment for these frogs. But it's sort of based on some biology for these animals. So in Minnesota, where these studies were conducted, it's not uncommon to find these two species sitting in relatively close proximity producing calls. So this is the American toad. These guys produce really, really long sequences of pulses. They might last 40, 60, 80 seconds per call. Right? So it's not uncommon to find situations where a tree frog male sitting near a toad is calling, and you get the situation where the call is overlapped. And what I'm illustrating here is that the temporal structure of the call is obscured in conditions of overlap. And so we asked the question, well, could the frogs use frequency differences to segregate the target signal from a captor? And so we designed an experiment based on the Mary Had a Little Lamb example, where we gave the frogs an attractive target, and we embedded it in a continuous stream of pulses. We're calling it the captor. That just went on and on and on. Behaviorally neutral. The frogs neither went to it or ran away from it. So it didn't affect the frogs either way. But the idea here was that we interleaved the pulses. So that when the target signal came on, the instantaneous pulse rate was 2 times the normal pulse rate, right, of 50 pulses per second. So the question is, what do they do with 100 pulses per second? Well, we know from control experiments that if they perceive something at 100 pulses per second, they hate it, they ignore it, they don't like it at all. And so the question was, well, if they integrate the target and the captor, right, then they should hear an unattractive sound. But if they're able to segregate the target from the captor, then the target's pulse rate of 50 pulses per second should emerge in this separate auditory stream. And so we varied the frequency separation between these two. And what I'm showing here is response latency. Now, I've played a cruel trick on you here. I'm showing a normalized response latency. So now fast responses are actually at the top. This is a slow response at the bottom. I'm just showing that as a function of frequency separation. And what you can see is that as the frequency separation between the target and the captor increased, the frogs behaved as if they increasingly recognized the signal and segregated it as a separate auditory stream. So our interpretation here is that frequency is one cue that they potentially can use in the natural environment. Okay, I'm out of time, but I'm going to just push a little bit. Very quickly tell you about one last experiment where we started looking at frequency and space together at the same time. Now, to understand this experiment, I have to give you a little background information based on previous work done with this species. We know that females, as I mentioned earlier, prefer longer sequences of pulses over shorter sequences of pulses, right? Females have a preference for longer calls. We also know that if you introduce gaps into a call, the frogs hate it, right? It shuts them off almost instantly if you put gaps in these sequences. So we decided to do this experiment where we traded off call length with the presence or absence of gaps. And we gave them sort of a long sequence of pulses that every other group of 5 pulses differed in frequency, right? So I'm labeling these A pulses and B pulses. And we gave them that choice versus an alternative of a short string of pulses that were coherent in space and frequency. Okay? And so if the frogs integrate the A and the B pulses together into one stream, they should perceive a long call that's more attractive than a short call. But if they segregate the A and the B pulses and they hear these gaps, right? They hate the gaps. So they should choose this shorter call, which is well within the range of the species, right? And then sort of the setup was like this. We gave them a choice either with the A and the B pulses from one location or from separated from 90 degrees. Does that make sense? So the results look something like this. And this is sort of a complicated graph, so let me sort of walk you through it really quickly. What I'm showing here on the y-axis is the proportion of females that chose that longer call with the A and the B pulses. And I'm showing here A frequency, and we've replicated the experiment twice. Once with the A frequency at that low spectral peak in the call, and once again with the A frequency at the high spectral peak of the call. And then we varied the B frequencies over the ranges I've indicated here. And integration corresponds to a high proportion of females choosing the longer call. And so what you find is that if we ask, does frequency help them segregate the call, the answer is it depends, right? It depends on the range of the frequency we're actually testing. So in this intermediate range between the two spectral peaks in the call, females were quite good at integrating A and the B tones. And this is interesting for two people in the room. Well, okay, at least one, possibly two people in the room. That's Peter. And this is actually integration occurring probably across different sensory organs in the frog's periphery, the amphibian and the basilar papilla. On the high end, when we varied frequency, response rates went down. And we can show that this just maps onto the audiogram. Right? So they're just not hearing the B pulses when we go high in frequency here. That's not the case on this end. In this end, this is well within the range of high sensitivity, but these frogs are still segregating based on differences in frequency, right? So as we move the A and the B tones away in frequency, they start preferentially choosing the short call. Okay? So what happens when we spatially separate the A and the B pulses? Again, the answer is it depends on frequency. And so here in the dashed lines, I'm showing the responses to the 90-degree separation. And the effects of frequency completely depended on whether or not the A frequency was at the high end or the B frequency was at the low end. And my graduate student and I are still negotiating about how to interpret this result, so I can't actually tell you what this means yet, but it's a stark result, and we're very interested in sort of following it up. So to summarize what I told you, looking at common spatial location, frequency separation, or frequency similarity as grouping cues in these frogs, this is our current sort of working interpretation, is that frequency is probably a more important cue than spatial separation. I don't want to rule out spatial separation just yet, but from work in other labs like Ham Ferris and Mike Ryan have done some other studies as well showing that these frogs are really willing to group sounds over large spatial differences, so it's probably not a real important grouping cue. And so this is sort of ongoing work we're still looking into. Now, if I have 45 seconds left, okay, if I have 90 seconds left, we'll wait till Eric says no. I want to sort of maybe wax a little philosophical for a little bit. And the framework I've been talking about for the experiments we're doing come from auditory scene analysis, right? This is a major paradigm in the studies of human hearing. And when you think about sort of the subdisciplines contributing to the study of auditory scene analysis, certainly human speech perception is important. There are a lot of people doing sort of basic human and animal psychoacoustics. There are plenty of people doing human and animal physiology. And there's an entire field that's developed called computational auditory scene analysis. And I have no idea whether this is something that people know about, but I thought if not, I would throw it up here. This is a really interesting book describing that. But what I often do in trying to get money from funding agencies is try to make the case that studying animal sequence perception is really important, right? That we as a field have something to contribute to what we know and understand about auditory scene analysis. Now, topic of this workshop is the analysis of animal vocal sequences. And this isn't at all a representative, completely representative sample, but you think about subdisciplines that may represent interests in the room. You know, there are plenty of people interested in sort of looking at algorithms for sequence analysis. I think a lot of us would be interested in sort of algorithms for automated signal detection and recognition. Certainly acoustic recording technologies are important for a number of people in the room. And then plenty of people look at sort of behavioral analysis of meaning from the receiver's perspective. But I would just add that sort of understanding how the animals that actually evolved to analyze vocal sequences would help us sort of flesh out the whole what do I want to say? Sort of the topic of how we study or how we approach the problems that we're interested in here. And then very lastly, I have a question, mostly for the computational people, and I'm just very curious as to whether there's any crosstalk between sort of what we're thinking about and what we're doing on the computational side and what sort of the human auditory scene analysis, computational auditory scene analysis folks are thinking about. There may well be, I just don't know, I'm curious. And so with that, I'll just acknowledge the people who actually did all the work I talked about today. Kason Reimers was an undergraduate in the lab, did some experiments, former postdoc Vivek Nidyananda, current grad student Katrina Schrotti, and if there's any time now or later for questions, we can take those. So, thank you.